This lecture is on the second topic in Unit 5. The second topic is immune system. The immune system is a defense system working inside our body, fighting against the foreign invaders, primarily microbes such as bacteria, virus, parasites, immune and system fungi. Is not just a cell, a single cell or a single organ. It is also a network of, it is a network of cells, tissues, various tissues and organs working together, fighting against the foreign. All the defense mechanisms working in our body can be classified into two types. One is the innate immune responses, and the second is the acquired or adaptive immune responses. Innate immunity is the defense system which is naturally present in an individual. The person is born with some defense mechanisms that is called as the innate immune responses. So these defense systems are present in an organism even before it is encountered with a foreign organism. That is why it is pre-existing. You are born with some defense system. That is why these immune responses are called as innate immune responses. And if you see these innate immune responses, they are non-specific. That is, the protection it provides is same no matter what the foreign invader is going to be. Innate immune responses are the first line of defense in an organism. When an individual encounters an antigen, the innate immune responses come into action first, preventing the entry of the foreign invaders. The innate immune responses consist of four types of barriers. 1. The anatomic barriers. 2. Physiological barriers. 3. Cellular barriers. And 4 are the inflammatory responses. Anatomic structures which are part of innate immunity are skin, mucous membranes and various other secretions. Skin, one of the anatomic barrier, is an effective mechanical barrier preventing the entry of foreign microorganisms. If you study the structure of the skin, it has an outer keratin layer, then a thin epidermis and a thick dermis. This dermis layer consists of hair follicles, blood vessels, sebaceous glands and sweat glands. If you see this sebaceous gland, it secretes a wax-like substance called as sebum. It consists of lactic acids and fatty acids, which helps to maintain a pH around 3 to 5. This low pH helps in inhibiting the growth of microorganism, thus preventing the growth and entry of the microorganism. Alimentary, respiratory and urogenital tracts are lined by mucous membrane. Cells in the mucous membrane, they secrete a viscous fluid which is called as mucus. Saliva, tears and other mucus secretions are all examples of this mucus fluid. What this mucus does is it entraps the invading foreign microorganisms and washes away from the membrane. Apart from that, the mucus fluid also has antibacterial and antiviral properties. In the lower respiratory tracts, there are hair-like protrusions on the epithelial cell membrane. What these hair-like structures does is they propel the mucus entrapped microorganism from these tracts. Entrapped microorganisms we throw out from the system by coughing or by swallowing it. Various physiological barriers that contribute to the innate immune responses are pH, temperature, and chemical mediators. The various soluble factors uh, contribute to the innate immune response. One is this lysozyme, which cleaves the bacterial cell wall, is present in the mucous secretions such as saliva and tears. So here naturally what we do when we have a cut, immediately we put that cut finger into our mouth. So naturally it is coming because that saliva has the antibacterial property. So saliva contains the lysozyme which helps in breaking down the bacteria which will enter through the cut. So it acts as a first line of defense. One of the other physiological barrier is the low pH of the stomach acid. So any bacteria that is swallowed is broken down by the strong acids in the stomach and preventing the growth of the bacteria. Next to the physical barriers, there are many cells and proteins involved in the innate immune responses. So mostly those cells are phagocytic cells, and the proteins like cytokinins are involved in the innate immune responses. The cells and the proteins, how they act against the antigens are, first they recognize the presence of the foreign organism, and then they provide an immediate cellular response to the foreign organism. The innate immune cells recognize pathogens by structures present on their cell surface. Here a phagocytic cell recognizes a pathogen by structures present on their cell surface. These cell surface structures, they are called as receptors. So they sense the foreign organisms, and usually these receptors are proteins or glycoproteins. And what they recognize on the pathogen is structures present on the surface of the pathogen or molecules secreted by the pathogen. Once the phagocytic cells recognizes, or the immune cell recognizes the pathogen, how they respond, how they act against it. So there are a number of ways in which they act against the pathogen. One widely seen method is the phagocytosis. What is this phagocytosis? So here the phagocytic cell, once it recognizes a foreign pathogen, it binds to it and extends the membrane projection around the bacterium and eats up or engulfs it. And inside the phagocytic cells, by strong hydrolytic enzymes, the engulfed bacteria is killed and degraded. So this entire process is called as phagocytosis. Various cells which take part in the innate immune responses are phagocytes such as macrophages and neutrophils, then dendritic cells, mast cells, basophils, eosinophils, natural killer cells, and lymphocytes. Dendritic cells, they are also phagocytic cells and they act as antigen presenting cells. Mast cells are involved in acute inflammatory responses such as asthma and allergy. Macrophages and neutrophils are phagocytic cells. The difference between them is macrophages have a very long lifetime. They can survive for several months to up to several years. And whereas neutrophils are also phagocytic cells, but they have a very short lifespan of 6 hours to few days. Eosinophils are other type of white blood cells which are involved in the immune responses. They help in acting against large parasites which cannot be phagocytized, and they are also involved in various allergic reactions. Basophils are cells which are involved in allergic reactions, and they are non-phagocytic cells. They are major effector cells in skin allergies. The next type of cells are the lymphocytes, especially the T cells, T lymphocytes. These cells act on intracellular bacteria. They activate cytotoxic and act upon virus-infected cells and tumor cells. Monocytes are the other cells, actually immature macrophages. So they differentiate into mature macrophages and involved in various activities. Natural killer cells are a type of lymphocytes, that is white blood cells. They are effective against working against cancerous cells and virus infected cells. These natural killer cells attach to the virus infected cells and release chemicals such as perforins and granzymes and destroy the virus infected cells or cancerous cells by secreting new proteins. These proteins can directly act upon the pathogens and kill the pathogens, or they can indirectly kill the pathogens by activating other type of cells to act against the pathogens. There are various proteins being secreted, but important group of proteins that are secreted in response to pathogenic stimuli are these cytokinins. The cytokinins are a group of small proteins which are involved in cell-to-cell communication to act against the pathogens. Some of the cytokinins are interleukins, colony stimulating factors, interferons, and tumor necrosis factor. Tissue damages due to infection or mechanical force or chemicals or extreme heat or cold induces a non-specific immune response called as the inflammation. The inflammation process starts when the tissue is damaged and release of chemical messengers such as histamine takes place. Once the chemicals are released, they expand the blood vessels, making it more leaky, allowing the immune cells and proteins to flow to the damaged area. The immune cells, such as phagocytic cells, act against the foreign particles entering through the damaged area. These phagocytic cells not only acts against the foreign microorganism but also act against the dead cells. and remove the debris. The main objective of the inflammatory response is to prevent the entry of the foreign particles through the tissue damage, clean up the damage, and start the repair process. In the inflammation region, you can see the skin is having slight swelling, redness, heat, and pain. Acquired immune response is the immune responses an individual acquires during his lifetime when he encounters a foreign pathogen. And the responses are very specific for each antigen encountered. Therefore, it is called a specific immune response. So, the acquired immune responses are capable of recognizing and selectively eliminating specific microorganisms and molecules. The basic characteristic feature of acquired immunity is antigen specific, and it is able to recognize billions of unique structures present on the foreign antigens. So it has diversity and has a memory of the antigen it has encountered. So it has an immunological memory and able to differentiate between self and the non-self cell and non-self. Adaptive immune responses consist of majorly two groups of cells. One is the lymphocytes, and the other type of cells is are the antigen presenting cells. Lymphocytes, they are type of white blood cells. These lymphocytes, they are produced in the bone marrow, and they circulate in the lymphatic system and the blood vessels, and they also reside in various lymphoid organs like thymus, spleen. So they also reside in organs, lymphoid organs. There are two major groups of lymphocytes involved in the adaptive immune response. They are the B lymphocytes, which is also called as B cells, and T lymphocytes, which are called as T cells. So these are the two major population of lymphocytes involved in the adaptive immune response. B lymphocytes, they get the term B because they are produced in bone marrow. They are produced and they get matured in bone marrow. Once it is matured, this picture is an illustration of a B cell. So you can see a cell. On its surface, it has some structures here, a receptor like structure. These structures, they are the B cell receptors. So what are these receptors do is they bind to the foreign particles, that is the antigens. They are cell surface receptors. Since they are present on the B cell, they are called as the B cell receptors, and they bind to unique antigens. So they are antigen binding receptors. And these receptors, if you see, they are membrane bound. They are spanning across the membrane. They are embedded in the membrane of the B cells. The B cell receptor, it is a glycoprotein, and it is called as immunoglobulin or antibody. If you see the structure of the immunoglobulin, it has 4 polypeptides. This is a glycoprotein. It is made of 4 polypeptides, 2 identical heavy chains and 2 light chains. 2 heavy chains and 2 light chains. So where it binds with the antigen is at this portion. This is called as the variable region where it binds to the. You see these immunoglobulins, that is the receptors protein which is present on the B cell. There are majorly of 5 classes. One is this immunoglobulin A, immunoglobulin D, immunoglobulin E, G and M. These are the 5 types of immunoglobulins. Immunoglobulin is a major secretory antibody. It is present in most of the secretions like tears, saliva, gastrointestinal fluid, milk and other mucous fluids. It constitutes about 10 to 15% of the serum. It is present in that concentration in the serum. Immunoglobulin D, it helps in stimulation of activation of B cells, and it is present in a very low level in the human serum. Immunoglobulin E are involved in allergic reactions. Immunoglobulin G, it is the only immunoglobulin which can pass across the placenta and provide immunity to the fetus and to the newborn infants. They are the most abundant immunoglobulins present in the serum. Immunoglobulin M, they are involved in the primary responses to an antigen. When an antigen encounters, the B cells can directly recognize the antigen and get activated to fight against it, or they recognize the antigen through the activated T helper cells and and the B cell gets activated and start acting against the when antigen. B cell encounters an antigen, it starts to proliferate, that is multiply in numbers, and differentiates further, that is it learns how to act against the antigen. So it recognizes, proliferates and differentiates into effector cells, effector B cells. The effector B cells consist of two population of effector B cells, where one group is a large group of effector B cells which are called as the plasma B cells, which will synthesize and secrete millions of antibody molecules. These secreted antibody molecules enter into the bloodstream, acting against the specific antigen which it has encountered. And a small population of the effector B cells remain as memory B cells. These memory B cells, they are in small numbers, and what is their role is, they have a very long lifespan and they are circulating, and when the individual encounters with the same antigen at a second time, these memory cells are activated and immediately brings about the response against the specific antigen. So here the immunoglobulin which has not encountered with any antigen, they have this receptor molecule bound on its surface, but when it becomes an effector cell, the plasma cell, this immunoglobulin or the receptor molecule which is bound on the cell membrane is secreted, that is it moves out from the cell and starts flowing in the bloodstream. And here each B cell makes one specific type of antibody for each antigen. That is, if a B cell encounters with a influenza virus, it will be producing antigen specific. All the effector cells will be producing antibodies specific for that influenza virus only. So each B cell produces antibody for one specific antigen. And if another B cell, if it is encountering with a chickenpox virus, the effector B cell from that B cell which has encountered the chickenpox, it will be producing antibodies specific for that chickenpox virus only. So each B cell will be producing antibodies specific for each antigen it has encountered. T lymphocytes are the lymphocytes which arise in the bone marrow. They originate in bone marrow but later migrate to another gland called as thymus, where they get matured. Once they get matured, they express the cell surface protein, which is called as the T cell receptor. These T cell receptors helps in recognizing and binding. But how they differ from the B cell receptors is the T cell receptors can recognize the antigens only when they are complexed with major histocompatibility molecules. So these T cell receptors can recognize antigen only when they are presented with the major histocompatibility complex, and they cannot recognize the antigens on their own. And when they encounter with an antigen, the T cells starts to proliferate and differentiate into effector cells. The effector cells, they are of two categories, where the major effector cells will be secreting proteins such as cytokinins, which in turn will go and activate other immune cells. And a small population of T cells, effector T cells, will remain as memory. There are some populations of T cells. They are T helper cells and TC cells, that is T cytotoxic cells. So here TC and TH, how you can differentiate majorly is based on this cell surface proteins. Here in the case of TH cells, they have these cell surface proteins called as cluster of differentiation 4, CD4. But whereas TC cells, they have the other type of cell surface proteins called as CD8. So what is the role of this TH cells and TC cells? TH cells initially recognizes the antigen which is being complexed with MHC molecules. Once it recognizes, the cell is getting activated and differentiated and become effector cells which secrete cytokinins. These cytokinins in turn will go and activate other cells such as B cells, TC cells, macrophages and various other immune cells. So here the effector TH cells, they also activate this TC cells, that is cytotoxic T cells. This TC cells, once activated, they will differentiate into effector cells secreting cytotoxic chemicals, or TC cells can also recognize antigens complexed with MHC class 1 molecules and get activated. Once activated, the TC cells, they proliferate, differentiate into effector cells which are called as the cytotoxic T lymphocytes. These TC cells can directly act against, once activated, they can directly act against the antigens. How they act is they release cytotoxic chemicals such as perforins, granzymes and granulysin, and they help in working against the killing the foreign microorganism. Since they kill the microorganism directly, they are having the cytotoxic property. That is why they are called as cytotoxic T lymphocytes. Antigen presenting cells are a major group of cells involved in adaptive immune responses. So here these are the cells which encounter the antigen initially. They engulf, they phagocytize the antigens and present the antigens on their cell surface by complexing it with class 2 MHC molecules. Some of the antigen presenting cells are macrophages, B lymphocytes and dendritic cells. The major property of this antigen presenting cells is they express this class 2 MHC molecules on their surface, and they also secrete signals which are necessary for Th cell activation. The adaptive immune response mediated by B cells are known as humoral immune responses. Since the B cells secrete antibodies which circulate in the blood, the immune responses are called as humoral, blood-mediated immune responses. The T cell mediated immune responses are called as cell-mediated immune responses. The different organs taking part in the immune defense system are broadly classified into primary lymphoid organs and secondary lymphoid organs. What are the primary lymphoid organs? They are thymus and bone marrow. So thymus and bone marrow are the primary lymphoid organs where the maturation of lymphocytes, that is B cells and T lymphocytes, take place. Thymus, one of the primary lymphoid organ, is a bilobed organ situated above the heart. It is the site where the T cells mature, develop and mature. The T cells, they originate in bone marrow, but they move to thymus where they develop and bone marrow, that is inside the bone you see a brown meshy structure. That is the bone marrow. So it is the site where the B cells originate and develop. Secondary lymphoid organs, they are lymph nodes, spleen, tonsils, mucosal associated lymphoid tissues. That is loosely arranged lymphoid tissues in intestinal, respiratory, urino and reproductive tracts have scatters of B cells, T cells and plasma cells. So this all together, that is called as mucosal associated. The secondary lymphoid organs are the regions where the antigens are entrapped. And these secondary lymphoid organs provides the site for the mature T and B cells to act against the trapped antigens. So secondary lymphoid organs is where the matured B and T cells migrate to and act against the trapped antigens.